So we have here the two pentose sugars that are responsible for our DNA and our RNA. Our DNA and our RNA, deoxyribose is responsible for DNA, deoxyribonucleic acid. Ribose is the pentose sugar in RNA, ribonucleic acid. The difference here is that deoxyribose, deoxy, is lacking in oxygen on the 2' carbon compared to ribose, which has a hydroxyl group here, an OH group here at the 2' carbon. So this is the only difference between these two pentose sugars. 2' hydroxyl group on the ribose sugar, 2' hydrogen on the deoxyribose sugar. That is the only difference. This difference results in increased stability of the deoxyribose molecule and allows it to form into these double helix structures. So, now let's talk about our nitrogenous bases. We have adenine and guanine, which are our purines, and we have cytosine, thymine, and uracil, which is only found in RNA, and thymine, which is found in only DNA, and these are our pyrimidines. Adenine and guanine, our purines, have a double ring structure. They have 2 rings. And our pyrimidines have a single ring. This means adenine can only pair with thymine and uracil, making 3 rings total distance between the chains of nucleic acid, And cytosine can only pair with guanine, 3 rings of distance between the 2 backbones of DNA. Now, no, you do not need to memorize the nitrogenous base formulas. I only want to drive home the point that purines have 2 rings and pyrimidines have 1 ring. However, I do need you to remember hydrogen bonding, remembering that it is a dashed line. There are two hydrogen bonds that form, between thymine and adenine. Also between adenine and uracil as well. And 3 hydrogen bonds that can form between guanine and cytosine. So 3 hydrogen bonds can form between guanine and cytosine. 2 hydrogen bonds can form between adenine and thymine or uracil. This is the difference between adenine and thymine and adenine and uracil. Can you tell? It's a methyl group, right there. Absent in uracil, present in thymine. Otherwise, they are exactly the same. So, in terms of base pairing, only certain combinations will be working. DNA to DNA, A to T, G to C, T to A, and C to G. Even though RNA is single-stranded, RNA can base pair complementarily with DNA, and also with RNA, if a single strand of RNA loops around itself, or when it is pairing with other types of RNA during a process later that we will discuss, translation, for example. So DNA when paired with RNA, adenine to uracil, guanine to cytosine, thymine to adenine, and cytosine to guanine. Remember, the DNA is in blue and the RNA is in red. That's why we can have thymine here and adenine here, but adenine to uracil here. RNA to RNA, A to U, G to C, U to A, C to G. RNA to DNA, A to T, because DNA is our blue and RNA is our red, A to T. G to C, U to A, C to G. The takeaway point is A to either T or U, depending on whether you are working with DNA or RNA, respectively. And G always pairs with C, and vice versa. C always with G. Now, for the sake of simplicity, when you are going to be drawing nucleotides, you can draw phosphate groups as a circle, bonds as sticks, pentose sugars, no matter whether they are DNA or RNA, as a pentagon, and your base pairs as some sort of arrow some kind of arrow stick. But you must label if they are adenine or thymine or guanine or some such. By convention, typically Cambridge's convention is that purines are longer. And they have a convex edge, and pyrimidines are shorter, and they have a concave edge. So this is either T or U, depending on the circumstance. All right? That's by convention. However, don't be surprised if you see it other ways. It has been done many different ways. So, some common spelling mistakes, things that are specifically no-nos on the Cambridge exam. Adenine is a nitrogenous base. Ribose is a pentose sugar. When you add adenine and ribose together, you get adenosine. Adenosine is the combination of adenine and ribose together. They are not the same thing. Adenosine is not the same as adenine. Do not confuse the two. Thymine is a nitrogenous base. It is THYMINE. It has nothing to do with thiamine. Thiamine is vitamin B1. Pyrimidines are cytosine, thymine, and uracil. Pyramids are found in Egypt and Mexico, most notably. Uracil, once again, has nothing to do with Duracell batteries. So, now that we've cleared that up, I would like to introduce you to a very handsome molecule known as ATP, my good friend. So, ATP is adenosine triphosphate. Adenosine, adenine plus ribose. Adenine plus ribose. Triphosphate, 1, 2, 3, tri. Mono, di, tri. Phosphate groups. So there are 3 phosphate groups and adenosine, adenine plus ribose. So, ATP is effectively the most important molecule you're going to be dealing with. ATP is the storage unit we use for pretty much every energy-intensive process required for life. It is not energy. I want to reiterate this. ATP is not energy. It is energy currency. You spend and expend ATP for energy. You break the bonds in ATP in order to obtain chemical energy. But it is not energy in and of itself. Mitochondria do not produce energy. Mitochondria produce and synthesize ATP in order to break that ATP to use and to utilize that for its chemical energy. Do not substitute ATP for energy when you are writing the exam. That is unacceptable. You will not get points. All right. So the point here for ATP is that there are anhydride bonds. These are your high energy bonds. These release an enormous quantity of energy when they are broken. Somewhere between 30 and 12 kilojoules per mole, depending on what source you do, depending on whether you're talking about just a bomb calorimeter or you're talking about certain different systems. But how are we going to get this sweet, sweet energy out of ATP? Hydrolysis. Hydrolysis is how we are going to extract this energy from the ATP molecule. The ATP molecule, these anhydride bonds, these phosphorus oxygen phosphorus bonds are going to be broken by hydrolysis. We add water. We add the hydroxyl group here, and we get a hydrogen ion here, and we are left with ADP and an inorganic phosphate. ADP plus PI. So, we start with ATP. We add water. We yield inorganic phosphate and looks terrible. Let's fix this. We add we get inorganic phosphate and ADP, as well as an additional hydrogen ion. So that is the resulting reaction that occurs here. We also get a significant amount of heat. All right? So here is our simplified ATP. This is our phosphodiester bond. Let's talk now about how we combine, how we add how we add, can we cut here, actually?
Yeah, that's a good idea.